In this topic, we're going to have a look at chromosome mutations. So by the end of this topic, you should be able to answer the questions, what is a mutation? What is aneuploidy? What's an example of aneuploidy? What is autopolyploidy? What is allopolyploidy? And then we're going to look at the difference between a somatic and gametic mutation. So what's a mutation? This is a spontaneous, unpredictable change in the genetic material. So in the previous topic, we discussed mutations. You should remember that a chromosome mutation is when the number or structure of chromosomes is changed. So what is a gene mutation? This is when the DNA is changed, so it results in a different allele being produced. Now, what is aneuploidy? Aneuploidy is the loss or gain of a single chromosome due to the non-disjunction of homologous chromosomes during anaphase 1 of meiosis. Now, the gametes are either going to have an extra chromosome, N+1, or they're going to be missing a chromosome, N-1. Let's have a look at what I mean. If there's non-disjunction during meiosis, you can see that one gamete will have an extra chromosome, N+1, whilst another gamete will be missing a chromosome, N-1. Now, if these gametes are fertilized with a normal gamete, have a look at the resulting number of chromosomes on the very right. So if the gamete N-1 mates with a normal gamete, the zygote formed will usually fail to develop. If a gamete N+1 fuses with a normal gamete, the zygote formed will have an extra chromosome. Now, if this extra chromosome occurs on chromosome 21, the child will have something called Down syndrome. So here you can see the extra chromosome 21 on the karyotype or karyogram. The fertilization of a genetically abnormal egg carrying an extra chromosome 21, you can see the yellow circle, fertilized by a normal sperm, represented by the green circle, produces an embryo with Down syndrome. So the condition is characterized by mental retardation and physical abnormalities, such as eye and face structure. The heart can also be affected. So this is an example of aneuploidy, where you've got an extra chromosome 21. Then you've got something called Turner syndrome, which is another example of aneuploidy. Have a look at this karyotype or karyogram of a person that has Turner syndrome. Can you identify what is different about it? It's missing an X chromosome from the father. So this results in distinct physical characteristics. For example, a short stature with a webbed neck. Now, the girl's ovaries don't work properly, which means that she doesn't menstruate. It can also lead to congenital heart defects. So this is an example of aneuploidy, where you are missing the X chromosome. Okay, what is autopolyploidy? Autopolyploidy is when an organism has more than 2 sets of chromosomes derived from the same species. So during mitosis, the chromosomes don't separate. So we call this non-disjunction. So they don't separate into different cells because the cytoplasm doesn't separate the cells, so there's no cytokinesis. Now, the gametes produced by the flowers are diploid, as you can see here, 2N. And when they fertilize, they form a tetraploid. So what you have is a diploid pollen and a diploid ovule forming a tetraploid. So in this picture, this will only happen if you have 2 diploid gametes meeting. So the nucleus doubles in size and the organism becomes larger and sturdier. So keywords to remember about autopolyploidy is more than 2 sets of chromosomes, same species. Now you've got something called allopolyploidy. This is when you have two or more sets of chromosomes derived from different species. So when the gametes fertilize each other, you get a hybrid. And this usually occurs in plants. So we're going to see how we get something called the common bred wheat due to this. So the difference between allopolyploidy and autopolyploidy is allopolyploidy is from different species, autopolyploidy is from the same species. So where did the common bred wheat come from? Well, the bred wheat is a hexaploid, so it's got AA, BB, DD. So how did this combination of three genomes arise? Well, if you have a look at this picture, you'll see that one plant has got the genotype AA and another plant's got the genotype BB. So these would have formed a hybrid with one another, AB. This then underwent chromosome doubling and it formed a what did it form? It formed a tetraploid wheat. Another hybrid was formed when the genotype ABD resulted. And then this underwent chromosome doubling, and it resulted in a hexaploid wheat, which we call our common bred wheat. Right, let's go through this diagram step by step so it makes sense. It's basically the same as the previous simple one. So what I want you to look at is the number of chromosomes as we go along. And think about what's happening at each step. Okay, so many years ago, the wild einkorn formed a domesticated einkorn due to genetic mutation. So it wasn't a chromosome mutation, it was a genetic mutation. Now, when the domesticated einkorn and wild goat grass gametes fused, a hybrid was formed. Now, the gametes that each of these produced was normal, so you had 7 coming from the one plant and 7 coming from the other plant. However, because they're from different species, you get hybridization occurring. So you get a sterile diploid plant, which is going to be different from both the parents. So it's sterile because the chromosomes don't match up to form homologous pairs. And it's kind of like if you cross a donkey with a horse, you get an infertile mule. This means that meiosis can't occur because there aren't any homologous pairs. So the way that this sterile diploid could spread is by asexual reproduction. So at some point there was chromosome doubling, autopolyploidy. This is when Emma wheat was formed. So have a look at how many AB chromosomes there are in Emma wheat. So the doubling of chromosomes means that the chromosomes can now have one with which they can match to form homologous pairs. So if there's another mutation, you get another variation of wheat. And this variation happens because there's sexual reproduction. For example, meiosis. So the Emma wheat can now hybridize with wild grass to form a sterile triploid. How do you think the sterile triploid can reproduce? It can reproduce sexually if it undergoes chromosome doubling, and these chromosomes don't separate, so there's non-disjunction. So it's going to form the common bred wheat. So have a look at how many chromosomes there are in the bred wheat. So this means that the common bred wheat's nucleus is bigger, so the cell is bigger and the plant is hardier. Okay, and finally, what are somatic and gametic mutations? Well, somatic mutation occurs in somatic cells, so those are the cells that are not gametes. An example would be cancer. In a gametic mutation, a mutation occurs during the meiosis that forms the gamete. So this means that when the zygote is formed, this mutation is present, and it's going to be passed on to the daughter cells. It also means that this mutation can be passed on to the next generation. Okay, in summary, here are several new terms that we have learnt in this topic, so it's a good idea to copy them down and make small flashcards from them. So what is a mutation? This is a spontaneous, unpredictable change in the genetic material. Remember that you get a chromosomal mutation and a gene mutation. Then you've got this word aneuploidy. This is the loss or gain of a single chromosome. So examples we looked at were Down syndrome, where there's an extra chromosome 21, and Turner syndrome, where an X chromosome is missing. Do you remember this word autopolyploidy? This is when you have more than 2 sets of chromosomes derived from the same species. And then allopolyploidy. This is when you have 2 or more sets of chromosomes derived from different species. And finally, a somatic mutation and a gametic mutation. So a somatic mutation is a mutation in cells that are not gametes, and a gametic mutation is when there's a mutation during meiosis of the gamete. And that concludes our lesson, the end.